I start registration.
Okay, very well. So I think with this, then if this is sufficiently big, we'll carry on that way. I'm not sure that if I'm moving into presentation format, it will not wreck the whole uh, the whole presentation. So thank you, Fershte, for inviting me to this webinar, and I think we're off to a very good start. I'm personally, even though I'm not involved in this field, I'm a very big fan of the concept of repurposing drugs. I think there's lots of stuff in the existing therapeutic arsenal that has lots of potential, which has not been discovered so far. Also because analytical methods in the past, when those compounds were developed, were not what they are today. And the level of understanding that we have of fundamental science is opening a new avenue. So I think this is a very exciting field. So Professor Arkin, you're involved into an exciting venture. So all the best with this. So let me move briefly with, as I understand it is customary, a few words about myself. Don't get scared by the number of lines on that slide. Other than running my own practice, which is essentially a shell to host my activities, I'm involved quite extensively in the field of vaccines. As after being a board member at CanSino Biologics, which is one of the Chinese companies involved into the coronavirus vaccine development, I transitioned into a part-time executive role there. Besides, I'm having board positions in various companies, and I'm not gonna detail all of them, but suffice to say that several are in the field of vaccines, such as Eurocin Vaccines in Sweden, which is involved in adjuvants for immunotherapy, Vaccitech in the UK, which is the licensee of the Oxford technology for all applications except the coronavirus one, which went to AstraZeneca, and Univercells, which is a technology company backed by the Gates Foundation, involved in the localization of the production of biologics to decrease costs and facilitate access, availability, and therefore affordability. And besides, I do a little bit of investment advisory on startup companies in the field of life science. The usual disclaimer, since I'm involved with so many companies, some of them being public, don't blame anything that I will be saying to any of the companies I'm involved with. If you need to direct your wrath on someone, it's me. And this is the points that we discussed during the preparation that could be of relevance, which is essentially, how do we, in the current context, assess the relative performance of the vaccines, but most importantly, what drives their acceptability for all the stakeholders, not just, of course, the regulatory authorities of the healthcare providers, but of course, increasingly relevant is the opinion of the end user, the subjects, the people who will be vaccinated. And I think the question behind this is, could technologies drive, in a way, a superior acceptability by stakeholders if they were answering some of their concerns? And let me start with a few introductory considerations and put things into context. It's a very simple and bird's eye view on the situation. Do we have a consensus on what is the criteria to gauge the utility and beyond this, the acceptability of the current COVID-19 vaccine candidates? I would argue that we don't have a consensus and if there was only one symptom of the absence of consensus, I would actually refer to the cacophony that ranges from the world of publications all the way to the world of lay media. But it seems that there is a growing understanding that the beginning of the solution is dealing with severe infections rather than all infections, which would be, I assume, particularly relevant for the emerging variants, the new variants, and of course the question of generating the right level of herd immunity. And therefore, as such, the indicators which are particularly relevant for the public health authorities are hospitalizations, even more so than fatalities. And this is why there's such a focus on occupancy of facilities, care consumption. It's kind of interesting that this infection, contrary to influenza, for instance, to compare with the previous pandemic back in 2009, this coronavirus infection is sending people to the hospital for longer, and you have sequelae that last also longer. So the average care consumption per subject is actually substantially bigger than what we have experienced with our healthcare systems in the past. And therefore, maybe a realistic goal is to turn the infection into a mild illness or an asymptomatic case, which will be therefore very similar to what we have with the other endemic coronaviruses. And with the expectation that if we prime the immune system properly, we will, elicit a sufficiently strong line of defense to at least be I would say, a disease mitigator, more so than a plain vanilla prevention. And we have already, the good news is we have vaccines, those that are currently being used, that are preventing severe infections, and therefore they are good public health tools as soon as they're safe and acceptable. And of course, as you know, the new question is, are they going to address successfully the new variants? And if no, then what is the need to initiate already the race for the second generation, which has already started in a number of companies, and not only the American companies. And I think we have to be realistic. I think there's an element of almost paranoia and at the same time fatigue with this, with the confinement measures or the lockdown measures that have been implemented on people. We will not be able to prevent all airborne diseases, although those measures have substantially curbed the level of infection with RSV, with influenza, and so on and so forth. But you've got a bunch of other things out there that are much less dangerous and lethal. And other than if you decide to live in an astronaut suit or in a cave all by yourself, the odds that are that you can't protect yourself completely against those airborne viruses. And all of this is happening, again, as I said, with a heated debate among scientists and politicians on the background of an unbelievable epidemic of fake news, which I would refer almost to an infodemia. There's an epidemic or pandemic of bad news or fake news and misperceptions related to vaccine benefits, reliability of the development pathway that has been followed, and so on and so forth, which has to be addressed if we want, from the public health perspective, to bring people to the right level of acceptability of the vaccine. Now, when you look at the, and I think this is the first point I wanted to underscore here, when you look at the technologies that are backing the current vaccine candidates, they are not new. They are not new. And sort of in going back in time, and those are the four technologies that I'm going to be discussing: reverse vaccinology, structural vaccinology, synthetic biology, and the use of adjuvants. And starting by the juniors, I mean, reverse vaccinology, which is used, which has been used extensively to start designing the coronavirus vaccine, is already 20 years old. Okay, it is the principle of identifying vaccine antigens from the genome of the pathogens. The first use was the development of the meningococcal B vaccine. Right, so I think the first point, the first message that you see coming is, even though these coronavirus vaccines were developed fast, it is important to remember that this is based on years of insights on those technologies, and that's only one. Move to structural vaccinology. It's also a 20-year baby. okay, which is a field that really emerged in 2007 when it was demonstrated that high throughput structural determination was possible. But it really emerged in the year 2000. So same age, if you will, as the previous one. Now, when you look at synthetic biology, which is the notion of using synthetic genes for vaccination or even cancer therapy, okay, you use like a DNA cassette or you use oligonucleotides, so whether this is RNA vaccines or vector vaccines that express the spike protein on the surface, this is a technology that was pioneered in 1986. 1986, I was just barely graduated from university. Okay, so I'm telling you, it was not yesterday, and so it's important people understand that this is again not new. RNA was tried for the cancer vaccines back since 2005, right? So it is true that those vaccines have not yet reached the light of registration and commercialization, but it's been tried quite extensively. And lastly, I think the case in point is adjuvants. Everybody knows that adjuvants raise all sorts of frowning on the social networks, but essentially it's a technology that goes back to the 1930s, with the invention of alum phosphate and alum hydroxide. And then you had all the emulsion-based adjuvants, and more recently other adjuvants are being tried, such as TLR agonists. So I think the point here is you had a lot of insights and knowledge emerging from all those years of honing those technologies and trying them against other pathogens. And all this came together also thanks to internet sharing of information on the sequence of the virus that coded the spike protein. You add on top of this a layer of evolution of the design of clinical studies with the so-called adaptive design, when you move seamlessly from phase 1 to phase 2, phase 2 to phase 3, and the principle of adaptive licensing as well, where you get the possibility to submit your dossier as it becomes available, and therefore you save time on the regulatory review process. All of this essentially combined gives a fantastic collapse of the development time of the vaccine. Mind you, people who have still doubts, knowing all those facts, that there could be some, I would say, skeletons in the cupboard with the fast development of the coronavirus vaccine should know that the mumps vaccine that was developed decades ago was developed in 4 years, and that the Ebola vaccine was developed in 3.5 years. And Ebola was much harder to do a clinical efficacy study, considering the nature of the disease and the endemicity. So again, I think we are in a situation where technology is bringing a lot of progress, which is not always recognized and acknowledged as being acceptable at least by the lay public. So I guess the real question, and I'm nearing the end of the presentation, the real question is, how do you drive this acceptability all along the value chain, all the way to the end users? So this is a classical matrix that looks at the so-called 4A analysis, which is what drives acceptability, availability, accessibility, affordability of vaccines across all stakeholders. And I will focus only on the acceptability column for the sake of today's presentation. Of course, at the authorities level, the acceptability is largely driven by clinical efficacy, safety, dose regimen, possibly cold chain requirements, and the risk-benefit balance that stems from it. From the payer's perspective, it's quite evident. It's a matter of price, but at the end user level, the core of the matter is, what is the perceived purity, meaning absence of risk, versus the individual benefit that I can get. And in the specific case of this coronavirus infection, there is still a perception at the level of some tranches of the population that the risk represented by the vaccine is higher than the risk represented by the disease, which explains the resistance, if you will. So some of those vacillators will probably get convinced by a combination of, okay, many people have tried before me and they didn't drop dead. And besides, I'm fed up, having restrictions with my travel and so on and so forth. So therefore, I'm going to do it. But we have to face the harsh reality that there will be a fraction of the population that will remain completely resistant to the very idea of immunizing, and that will therefore represent a pool of susceptible subjects, which therefore create the possibility of some form of epidemic flare-ups, possibly localized in some geographies. So, if I was to summarize in an easy and I would say understandable way to the lay public, the balance of strength and unknowns rather than weaknesses of the three main technologies that are available today. So the RNA vaccines, time to market is definitely the main strength. Okay. They are increasingly demonstrating efficacy. We will have to see how long it lasts. Safety, as you know, there are some question marks. It looks like as usage is expanding, there's more reassurance about the fact that it is safe, even in patients with a profile of being prone to allergies. The cloud on the horizon will likely remain because this is again the first time that RNA-based products are being licensed and used in real life in the population. Viral vector vaccines, the strength is that you have probably more options relative to the previous ones. Formulation is also less of an issue. Time to market is kind of okay, even though it's a bit longer than the RNA vaccines. And you've got extensive clinical experience with these, more so than with RNA. So I think, on average, this compares favorably. The unknowns being capacity. RNA, you can produce a lot in a relatively small facility, whereas viral vector vaccines, you need, more sizable facilities, therefore a bigger capital investment. And there's the big question of whether you could boost it, by the way, which would apply also to the others. And as you know, there are the protein-based vaccines are coming up. There are some interesting results published recently by Novavax. There's extensive experience with protein-based vaccines, and this demonstration is also coming with the benefits of being adjuvanted if need be. So the bigger unknown with protein-based vaccine is the time to craft the vaccine and to bring it to market. But otherwise, there's a wealth of experience out there, and those would probably be therefore better accepted. So in a way, you could almost assume that you have from top to bottom an increasing gradient of acceptability if the primary driver was previous experience. But then, of course, the thing will average out over time because RNA vaccines being the first to be used large scale, there's accumulating evidence as we speak, both in terms of efficacy and in terms of safety. And hopefully over time we'll have the demonstration. I guess I would like to conclude on a note, which is that the proof being in the pudding. We'll have to see what happens in Israel with the coverage, because it looks like almost this country is going to be almost a real life clinical study in itself. This will be probably the first country where herd immunity could be demonstrated, and all eyes are therefore focused on what happens there and the evolution of the epidemiology. And I think we have to hope, in the interest of the population and public health, that we'll be able to bring a solution, an agile solution that will address the variants, and an acceptable solution so that we don't leave too many quadrants of the population susceptible, Otherwise, the problem will linger and become endemic in the worst possible way. And I guess on that note, I'm happy to take any questions if there were any.